All right, strap in, everyone, because today we are going deep, like way down into the cellular level.
Really gonna get our hands dirty with this one.
Yeah, we're talking RNA editing, ribosomes, protein synthesis, the whole shebang.
The works. All from some pretty intense academic notes.
Definitely not light reading, that's for sure. But if you're the kind of person who loves getting into the nitty-gritty, the actual nuts and bolts of how a cell makes its workhorses,
The proteins that do, well, basically everything.
then this is the deep dive for you.
Absolutely.
So should we just jump right in?
Let's do it.
Okay, first up, RNA editing.
Right. And the notes here really highlight guide RNA, gRNA.
Guide RNA.
Yeah, think of it like it's basically a blueprint that guides the whole editing process.
Okay, so it tells the cell what to change.
Yeah, exactly. And they actually give a really clear example in the notes. An edited RNA sequence ends up gaining 4 uracil nucleotides, right?
Uracil, that's one of the RNA bases.
Yep. And it gets those extra uracils because the guide RNA had 4 extra adenines.
Adenine, that's another one of the bases. But in the guide RNA.
Right. So you can see how that guide RNA is directly affecting the final RNA sequence.
Huh, so it's like a one-to-one thing. 4 extra adenines in the guide RNA means 4 extra uracils in the edited RNA.
Pretty much. And, the implications of this are huge because a single RNA molecule, it can be edited in all sorts of ways.
Meaning you get a bunch of different proteins from the same starting point?
Exactly. Like alternative splicing, but even before you get to the protein.
Wow, okay, that's cool. So then what about deamination? The notes mention that too.
Oh, yeah, deamination. That's where things get really interesting because that initial mRNA sequence.
It's not always set in stone, right?
Nope, not at all. Deamination can happen spontaneously, just by chance, or it can be done on purpose.
On purpose, like the cell makes it happen.
Right, with enzymes. But either way, it's a chemical reaction where you're basically removing an amine group from a molecule.
And a mind go.
NH2. It's a nitrogen and 2 hydrogens.
Okay, so it loses that. And the notes talk about 2 main types, right? Adenine and cytosine.
Yep. So adenine, when it gets deaminated, that turns into inosine.
Inosine, is that another one of the bases?
It is, but the thing with adenine deamination is it's often repaired.
The cell fixes it.
Yeah, like
Yeah.
it's got mechanisms to keep its genetic code clean.
Ah, okay, so that's like a safety net.
Exactly. But then you've got cytosine deamination. And that's got some really big implications, evolutionarily speaking.
Well, how so?
Well, when cytosine gets deaminated, it becomes uracil.
Right.
Now, this is a big deal because DNA uses thymine instead of uracil.
Yeah, why is that? They seem pretty similar.
Well, think about it. If DNA used uracil and then a cytosine got deaminated.
It would also become uracil.
Exactly.
Oh.
And then how would the repair machinery know which uracil was supposed to be there and which one came from a cytosine?
Oh, I see. So by using thymine, DNA avoids that whole problem.
Right. It's like a built-in way to make sure DNA repair is super accurate.
That's so clever. And there's that example in the notes, right, about ApoB100 and the small intestine.
Yeah, so ApoB100, that's involved in lipid transport. But in the small intestine, a specific cytosine gets deaminated.
And it changes the protein.
Big time. It actually turns a CAA sequence into UAA,
and UAA.
a stop codon. So instead of making the full ApoB100 protein, you end up with a shortened version called ApoB48, all because of that one little deamination event.
Wow, that's crazy how such a small change can have such a big impact.
Absolutely. It's a classic example of a nonsense mutation caused by deamination.
Okay, so we've talked about RNA editing, we've talked about deamination. Now what about those protein synthesis machines? The ribosomes.
Ah, the ribosomes. The notes actually start by talking about this guy, Masayasu Nomura, spent years and years purifying these things.
Purifying ribosomes. Why?
Because he wanted to figure out what they were made of, and his work was revolutionary, really. He showed that ribosomes are mostly RNA.
Wait, really? I always thought they were mostly protein.
Nope, it's RNA with a smaller protein component. And that was a huge deal because back then, everyone thought proteins were the only things that could catalyze reactions in the cell.
So basically, ribosomes are RNA machines that build proteins. That's mind-blowing.
It is pretty wild when you think about it. So want to get into the structure of these things. The notes have a whole section on E. coli ribosomes.
Yeah, let's do it. Break it down for me.
Okay, so E. coli ribosomes, like all ribosomes, have 2 subunits. The small subunit, called 30S.
30S. What does the S stand for?
Svedberg units. It's a measure of how fast a particle sediments when you spin it really fast in a centrifuge.
Okay, so 30S.
And the large subunit, 50S.
50S. So together that's 80S.
Actually, no. That's where things get a little tricky. The
Yeah.
Seedberg unit isn't directly additive because it relates to the logarithm of a particle's weight. So a 30s subunit and a 50s subunit together make a 70s ribosome.
Okay, I get it. Logarithms, always messing things up.
Right. But anyway, the 30s subunit is made of 21 proteins and one rRNA molecule, 16 srRNA.
16S, got it. And a large subunit.
That one's got 36 proteins, but 3 of them are repeated, so it's really 33 unique ones, plus 2 rRNA molecules, 5S and 23S.
Okay, that's a lot to remember. So all those proteins and RNA molecules come together to make this protein building machine.
Exactly. And you know what's really cool? Ribosomes are self-assembling.
Self-assembling, what does that even mean?
Like you can take them apart, denature them, separate all the pieces.
Unfold them completely.
Yep. And then, under the right conditions, they'll just spontaneously put themselves back together. It's like they have this innate blueprint for how to assemble.
That's incredible. Imagine the complexity of those structures,
Yeah.
and yet they can just build themselves.
Right. The notes actually have this cool visual representation of a ribosome. The rRNA is shown as these solid areas, and the proteins are more filamentous.
So it's like the RNA forms the core, the framework.
Yeah, pretty much. And all the really important catalytic sites, like where the tRNAs bind and where the peptide bond forms, those are all made of RNA.
That's more evidence for the RNA world hypothesis, right?
Oh, absolutely. The fact that RNA can catalyze such crucial reactions, it really points to RNA being a major player in early life.
Yeah, that idea that RNA came first, able to both carry genetic information and do stuff.
Exactly. Replicate, catalyze reactions, all that.
Wow, okay. And then, let's see, what else about ribosomes? Oh, the notes mention that prokaryotic and eukaryotic ribosomes are surprisingly similar.
Oh, yeah, that's super interesting. Like, you'd think with all the differences between bacteria and, us.
That their ribosomes would be totally different too.
Right. But no, they're remarkably similar in both their secondary structure and their overall composition.
Huh, so why would that be?
Well, it probably means that translation, the process of making proteins, it's already so finely tuned, so optimized that there's just not a lot of room for variation.
Like it's already so good, evolution can't really improve it much more?
Exactly. And that actually makes ribosomes a really interesting target for drugs.
Drugs? How so?
Because if they don't change much over evolutionary time, it's harder for bacteria to develop resistance.
Oh, I see. So if you target something that's essential and doesn't change much, you're less likely to run into resistance problems.
Right. It's a whole area of research, actually.
That's fascinating. So much to think about.
There is.
All right, so we've covered RNA editing, we've tackled ribosomes. Now should we move on to the main event,
Protein synthesis itself.
the big show? What do those notes say about the steps involved?
Okay, so they break it down into 5 main steps. First, you have amino acid activation.
Amino acid activation.
Right. Then there's initiation, where the ribosome gets everything set up.
Initiation. Okay.
Then you have elongation, where the polypeptide chain actually starts to grow.
Elongation. Got it. What's next?
Termination, where the whole process comes to a halt.
Termination.
And finally,
Protein folding. That's where the newly made polypeptide chain contorts itself into its final functional shape.
so those are the 5 steps. Amino acid activation, initiation, elongation, termination, and protein folding. You got it. And the notes even have this super detailed table breaking down all the components needed for each stage in E. coli.
Oh, wow. So for people who really want to get into the weeds, this is it.
This is it. It's a goldmine of information, but maybe we should take it one step at a time.
Good idea. Let's start with that first step, amino acid activation. What's going on there?
Well, that's where the tRNAs come in. They're like the couriers, delivering the amino acids to the ribosome.
Right, tRNAs, those are the little cloverleaf-shaped molecules, right?
Exactly. Cloverleaf shape. And they've got 4 main arms.
4 arms.
Yeah. You've got the amino acid arm, where the amino acid attaches. Then there's the anticodon arm, and that one carries the anticodon.
The anticodon, that's what base pairs with the codon in the mRNA, right?
Right. It's like a lock and key system. Each anticodon matches up with a specific codon, ensuring the correct amino acid gets added to the growing protein.
So it's all about making sure the right amino acid is delivered at the right time.
Precisely. And it's amazing how the 5-foot end of the anticodon interacts with the wobble position, that third nucleotide of the codon.
The wobble position.
Yeah, it allows for some flexibility in the base pairing, which is why multiple codons can code for the same amino acids.
That's the degeneracy of the genetic code, right?
Right. Multiple codons for the same amino acid. Now, the other 2 arms of the tRNA, those are the D arm and the tRNA.
D-arm, and TC arm. Okay. And what do those do?
Well, they're involved in the folding of the tRNA and how it interacts with the RNA in the ribosome because remember, these tRNAs aren't actually flat clover leaves in the cell.
Oh, right. They have a more 3D shape.
Exactly. It's more like a twisted, kind of screw-like structure, and that spatial arrangement is really important.
In what way?
Well, it makes sure the TC and D arms are close together, which lets the anticodon stick out, you know?
So it can easily base pair with the codon.
Exactly. Efficient codon recognition. Okay, but so far we've only talked about the tRNA. How does the amino acid actually get attached to it in the first place?
That's a good question. It can't just magically appear there.
Nope. There's a whole other set of players involved. Aminoacyl tRNA synthetases.
Aminoacyl tRNA synthetases. Try saying that 3 times fast.
I know, right? But seriously, these enzymes are incredible. They're like the matchmakers of protein synthesis.
Matchmakers.
Yeah, because their job is to make sure that each amino acid is attached to its correct tRNA, and they do this with incredible specificity.
So there are 20 different aminoacyl tRNA synthetases, one for each amino acid.
Yep, 20 different ones, and each one recognizes both its specific amino acid and the correct tRNA. It's a two-step process, and it requires energy from ATP.
So ATP is providing the energy to make that connection. It's not just attaching the amino acid, but also like storing energy for later.
Exactly. It's like prepaying for the next step in protein synthesis. And there are actually two main classes of these synthetases, class 1 and class 2.
Class I and class 2, what's the difference?
They have different mechanisms, different ways of attaching the amino acid to the tRNA. Class I attaches it to the 2' carbon of the ribose first, and then moves it to the 3' carbon.
Okay.
Class 2 just goes straight for the 3' carbon, different routes, same destination. And this whole interaction, the synthetase recognizing the tRNA and the amino acid.
It's like another layer of specificity on top of the codon-anticodon pairing.
Exactly. And this is sometimes called the second genetic code.
The second genetic code.
Yeah, because you've got the first genetic code, which is a codon specifying the amino acid.
Right.
And then you've got the second layer where the synthetase makes sure the right amino acid is actually attached to the right tRNA.
So it's like a double-checking system to prevent errors.
Exactly. Because, the ribosome itself, it can't actually check if the right amino acid is attached to the tRNA.
So if a mistake happens here, the wrong amino acid gets incorporated into the protein.
Yep. The notes even mention this example a la tRNA synthetase. It's got this really simple recognition mechanism based on a GU base pair.
GU base pair.
Yeah. And even if you remove a bunch of nucleotides from the tRNA, it still works. It can still recognize its target.
Wow. That's pretty amazing. But I think we're starting to get into the weeds a bit here. Maybe we should save the rest of amino acid activation for next time.
Sounds good to me. There's a lot more to unpack, that's for sure.
We've got initiation, elongation, all that good stuff still to come. But for now, we'll leave it there.
Yeah, let's give everyone a chance to digest all this.
Definitely. This is definitely a deep dive, and we're just getting started.
Welcome back, everyone. Ready to dive back into the depths of protein synthesis.
I'm ready to elongate my knowledge. Get it? Elongation.
Very funny. But yeah, elongation, that's where the real action is, where the polypeptide chain actually gets built.
Okay, so remind me, where did we leave off? We had the ribosome all set up, right?
Right, we had the initiation complex formed, the mRNA is bound, the initiator tRNA is in the P site.
And the A site is empty, waiting for the next amino acid tRNA to come along.
Exactly. So picture the ribosome like, think of it as a little factory.
Okay, a ribosome factory.
And it's got these 3 stations or binding sites for tRNAs. The A site, that's where the incoming aminoacyl tRNAs arrive.
They bring in the raw materials, the amino acid.
Exactly. Then you've got the P site, that's where the growing polypeptide chain hangs out, attached to the tRNA that brought in the previous amino acid.
Okay, so the P site is like the assembly line.
Yeah, I like that. And then lastly, you have the E site. That's the exit. The tRNA that's done its job, it goes there and gets kicked out of the factory.
So A site is for arrival, P site is for polypeptide, E site is for exit. Got it. So how does the right aminoacyl tRNA, the one with the next amino acid in the sequence, how does it know to go to the A site?
Well, that's where the elongation factors come in. EFs.
EFs.
Elongation factors. They're like, I guess you could think of them as delivery trucks.
Delivery trucks for aminoacyl tRNAs.
Exactly. In bacteria, the main one is called EF2. Eukaryotes use EEF1A. But the point is, these EFs, they bind to the aminoacyl tRNAs and bring them to the ribosome.
Okay, so they're like the chaperones, making sure the tRNAs get to the right place.
Right, but not just any tRNA can bind to the A site. It has to be the one with the anticodon that matches the codon on the mRNA.
Oh, right. Codon-anticodon pairing. That's how the ribosome knows it's getting the right amino acid.
Exactly. It's all about that specificity, making sure the genetic code is translated accurately. So let's say the right tRNA has made it to the A site. What happens next?
Well, I guess the amino acid needs to be attached to the growing polypeptide chain, right?
You got it. And that's where the magic of peptidyl transferase comes in.
Peptidyl transferase. Sounds important.
It is. This is the enzyme that catalyzes the formation of the peptide bond between the amino acids. And you know what's really cool about peptidyl transferase?
What?
It's not a protein. It's a ribozyme.
A ribozyme. You mean it's made of RNA?
Yep. It's actually part of the 23S rRNA in the large ribosomal subunit. So it's RNA catalyzing the formation of the peptide bond. Pretty amazing, right?
That's wild. I always thought enzymes were proteins. This means RNA is directly involved in building the polypeptide chain.
Exactly. Another piece of evidence for the RNA world hypothesis, but let's get into how this actually works. So the amino group of the amino acid on the A site tRNA.
It's attacking the carbonyl group of the amino acid on the P site tRNA.
You got it. And that forms the peptide bond. So now the growing polypeptide chain, it gets transferred from the tRNA in the P site to the tRNA in the A site.
Okay, so the chain is switched places.
Yep. The P site tRNA is now empty, and the A site tRNA is holding the elongated polypeptide chain.
So the chain is getting longer, one amino acid at a time.
Exactly. But now the ribosome needs to move down the mRNA, right? So that the next codon can be exposed in the A site.
Right, it's like the ribosome is reading the mRNA codon by codon. But how does it move?
That's called translocation, and it requires another elongation factor, EFG in bacteria, EEF2 in eukaryotes.
More elongation factors.
Yep, these guys bind to the ribosome and use energy from GTP hydrolysis to basically nudge the ribosome forward by one codon.
So it's like a little molecular motor powered by GTP.
Exactly. And during translocation, the tRNAs move too. The A site tRNA with the polypeptide chain, it shifts to the P site.
And the empty P site tRNA.
That one moves to the E site and gets ejected. So now the A site is empty again, ready for the next aminoacyl tRNA, and the whole cycle repeats.
Wow, it's like a perfectly choreographed dance with all these molecules moving in sync.
That's a great way to put it. And the cycle just keeps going, codon by codon, amino acid by amino acid, until...
Until the ribosome hits a stop codon.
You got it. That's the signal to end the whole process.
And we're back for the grand finale. We've made it from DNA to RNA to a full-fledged polypeptide chain. But there's one last crucial step.
The transformation from a linear chain of amino acids to a fully functional three-dimensional protein. We're talking about protein folding.
The protein's got to get all origami on us. But seriously, how does this happen? I mean, the chain's just floating around, right? How does it know how to fold itself into the right shape?
Well, it's often called a spontaneous process.
Spontaneous, like it just happens.
In a way, yeah. For many proteins, the information needed for folding is already encoded in that amino acid sequence.
Like a built-in instruction manual?
Pretty much. But it's not just about the sequence itself. There are all these forces at play that help shape the protein into its final form, like a molecular sculptor working on its masterpiece.
Okay, so what kind of forces are we talking about here?
Well, one of the big ones is hydrophobic interactions.
Hydrophobic, meaning water-fearing.
Exactly. So those hydrophobic amino acid side chains, they don't like being exposed to water.
So they try to hide.
Basically, they tend to cluster together on the inside of the protein, away from the watery environment. And that actually drives a lot of the initial folding.
Oh, I see. So it's like the hydrophobic parts are trying to get away from the water,
Yeah.
and that forces the protein to fold in on itself.
Exactly. And then you have hydrogen bonds forming.
Hydrogen bonds, those are the weak bonds.
They're weaker than covalent bonds, yeah, but they're still really important for protein structure. They form between the backbone atoms of the polypeptide chain and also between some of the amino acid side chains. They help stabilize the protein and contribute to its specific shape.
Okay, so I've got the hydrophobic interactions pushing the protein to fold, and then the hydrogen bonds helping to hold it in a specific shape.
Right. But there's more. You've also got electrostatic interactions.
Electrostatic meaning charges.
Yep. Some amino acid side chains have positive charges, some have negative charges.
And opposites attract, right?
Exactly. So those oppositely charged side chains will be attracted to each other, and that can influence how the protein folds.
Like charges repel.
Right. So those will push each other apart. It all contributes to the final 3D structure. And we can't forget van der Waals forces.
Van der Waals forces, those are.
They're very weak attractions between atoms, but there are a lot of them, so they do contribute to the overall stability of the folded protein.
Okay, so we've got this whole orchestra of forces all working together to shape the protein.
A very delicate and intricate dance. But we should mention that not all proteins can fold all by themselves.
Oh, what do they need help with?
Well, some proteins, especially larger ones with more complex structures, they need chaperones.
Chaperones, like molecular chaperones.
Yeah, exactly. These are special proteins that help other proteins fold correctly.
So they're like protein folding assistants.
Pretty much. They bind to the unfolded protein, prevent it from sticking to other things incorrectly, and help guide it along the right folding pathway.
So they're like making sure the protein doesn't make any wrong turns.
Exactly. And they often use energy from ATP hydrolysis to do their job.
So even in the world of protein folding, you can't escape the need for energy. But why is all this so important? Why does the protein have to fold correctly?
Well, a protein's function is directly related to its shape. If it doesn't fold correctly, it might not work at all. Or worse, it could actually cause harm.
Oh, so misfolded proteins are bad news.
They can be, yeah. A lot of diseases are actually linked to misfolded proteins, Alzheimer's, Parkinson's, things like that.
Wow, I didn't realize protein folding was so important for our health.
It is. And there's still so much we don't know about it. It's a really active area of research.
Well, I think our listeners' brains are probably pretty full of information by now.
We have covered a lot of ground, that's for sure.
From RNA editing to ribosomes to the entire process of protein synthesis, and now even protein folding.
It's amazing to think that all of this is happening inside every cell of our bodies all the time. Trillions of tiny molecular machines working together to keep us alive.
It's pretty mind-blowing when you think about it.
It is, and it's a reminder that there's always more to learn, more to explore. The world of molecular biology is full of these incredible stories just waiting to be uncovered.
So to our listeners, keep asking those questions. Keep diving deep. There's a whole universe of knowledge out there just waiting to be explored.